Our today's topic is peptides and proteins. Now, in biochemistry, the term protein is non-specific because it includes amino acid chain of any length. However, polypeptides refer to the proteins of a particular size. Still, we are able to define you certain these terminologies as far as the peptides and proteins are concerned. Remember one thing, that peptides, polypeptides and proteins, they are polymer of 20 standard amino acids linked together by peptide bond. So there is a requirement. The building blocks of these peptides and proteins are amino acids, 20 standard amino acids. Standard amino acids are those amino acids which have got a genetic code in the form of codon on the messenger RNA. One codon code for one amino acid. So only standard amino acids, they participate in the synthesis of peptides, polypeptides and proteins, and the only covalent bond which is present in these structure is the peptide bond. A compound is said to be oligopeptide when it is comprised of 2 to 10 amino acids. When the two amino acids they link by peptide linkage, they are called dipeptides. When 3 amino acids are linked by peptide linkage, they are called tripeptides. Simultaneously, the 4 amino acids made tetrapeptides. A polypeptide is made up of more than 10 amino acids. Now here we will give you a definition of peptides. The compound obtained by condensation of up to 10 same or different alpha amino acids, they are called peptides. Peptides are relatively shorter chains of amino acids, and most of these peptides are being generated from the breakdown of the larger proteins. As far as the example of the peptide is concerned, we have many examples like hormone, antidiuretic hormone, oxytocin, glutathione. Now polypeptides, they contain 40 or more amino acid unit, then they are called polypeptides. And remember, as I told you, proteins are polymers of amino acids of any chain. So all proteins are polypeptides. But a polypeptide is a long unbranched chain. Now this here I am stressing upon the word long and unbranched, no branching of the amino acid. And number two, they have a molecular weight of less than 10,000 daltons. So remember one thing, that polypeptides, they have 40 or more amino acids. They are unbranched chains of long unbranched chain, and they have a molecular weight less than 10,000 dalton. And the examples of the glucagon and the insulin. Now here you can see the amino acids, single amino acids. Then when up to 10 amino acids, they are linked by the peptide linkage. You can see this mediated structure linked by the peptide linkage, then they are called peptides. And you can see here, it is, you can't see any branching over here. They are linear unbranched small polymers. And here you can see a protein structure which is comprised of amino acid chain of any length. And you can see, when we say proteins, then they are being assuming their three-dimensional conformation, and being that structure is being stabilized by not only by the peptide bond, but also it includes some non-covalent bonds like ionic interaction, hydrophobic interaction, van der Waals forces. Then you are very much familiar that disulfide bonds are observed. So protein is a compact three-dimensional structure which is not a linear unbranched structure. It undergo folding and assume a three-dimensional structure, and beside peptide bonds, other non-covalent bond as well as the disulfide bonds are involved in stabilizing the structure of protein. Now here is an example of a dipeptide. We have one amino acid alanine and another is serine. And when the two are linked by peptide linkage, they form an allyl C. Now remember one thing, when an amino acid is being incorporated into a peptide, polypeptide or protein, we call them not amino acid, we will use the word amino acid residue. So you can see two amino acid residues, they are forming a dipeptide anyl. This is a very important tripeptide, that is glutathione, and glutathione is comprised of glutamate, cysteine and glycine. A tripeptide which is of a very important biological significance, and we will discuss the significance of glutathione while going through. Now these are also included now till 10, if you recall, oligopeptides or peptides up to 10. So these are the two important posterior pituitary hormones, oxytocin and antidiuretic hormone, and they are made up of 9 amino acids linked by peptide linkage. So oxytocin and vasopressin. Vasopressin is the another name of the hormone, antidiuretic hormone. Glucagon. Glucagon now till this vasopressin and oxytocin, they were peptides. Now here you can see the number is more than 10, so it is glucagon. It is again a hormone secreted by the pancreas. Glucagon, it is a peptide hormone. 29 amino acids are there, and you can see the linear chain structure, no branching you can see over here, and their molecular weight is less than 10,000. Insulin. Now insulin is made up of two polypeptide chains, and you can see here the only beside peptide bond, the another bond which is being observed over there, you can see three dipeptide linkages are being observed. So these dipeptide bonds, they link two peptide chains together, and active insulin structure is being constructed. This slide will enable you to differentiate a protein from polypeptide. Now, as we have already discussed that polypeptides, they are long, unbranched chain of amino acids, and they are made up of, their building block is amino acid, their molecular weight is less than 10,000 daltons, and the bond which is present in peptides or polypeptides is only peptide bond. Whereas protein is a complex structure, and it is made up of large number of amino acids. Here we have a limit in case of polypeptides, but in proteins unlimited number of amino acids, number one, and it is a complex structure. Number two, it is made up of polypeptides. So proteins are made up of polypeptides, and their molecular weight is more than 10,000 dalton. Here I will just correct it. And beside peptide bonds, the other non-covalent bonds are also present, like disulfide bond, ionic bond, van der Waals forces, hydrophobic interactions. So this will enable you to differentiate a polypeptide from a protein. But remember, proteins, all polypeptides are protein. Now these are the few examples of the few physiologically active peptides तक हमने जो डिस्कस किया था from 2 to 10. Okay, so the examples of few peptides, we won't be able to discuss each and every peptide in this lecture, but we will discuss important peptides which have important role, biochemical role in the body. One is glutathione, then we have bradykinin, then we have angiotensin, hormone vasopressin, oxytocin, thyrotropin releasing hormone, and methamphetamine. So we will go and discuss each peptide one. This is the structure of glutathione. We have already discussed that it is a tripeptide, and it is comprised of glutamate, cysteine and glycine. When amino acids, they are linked by peptide linkage, they are called amino acid residues. So now this glutathione is called as glutamyl cysteine glycine, a tripeptide of a biological significance. And in cysteine, if you recall, cysteine is a sulfur containing amino acid, and it has got a SH group that is called as sulfhydryl group or thiol group. And this SH group, it can be in the reduced form, a thiol group, or it can be oxidized form. So the reactive portion of the glutathione is its thiol group. And like this transitional metals like iron, it may undergo oxidation and reduction. Okay, it can accept electron and it can donate electrons. So it is being again utilized. It is a requirement of the oxidation reduction reaction. The reduced form of the glutathione is written as GSH, and when it is in the oxidized form, it can be written as GSSG. Okay, so it can be like iron in ferric or ferrous form, it can be in the reduced form or in the oxidized form, and it is a requirement for this oxidation reduction reaction, and it is being required to. Now if you recall in this iron this when we were discussing heme, that iron can accept or donate electrons, and it is been a part of the oxidation reduction reaction, and heme is a prosthetic group for catalases and peroxidases. So the enzyme peroxidase require reduced glutathione to be functionally active. Xenobiotics are actually the chemical compounds which are foreign to the human bodies, like drugs, pesticides, food additives. Bradykinin is another peptide, and it is being released in the body from mast cells when a patient is suffering from an asthmatic attack, or it may come from the gut wall or from the damaged tissues of the body. So bradykinin causes vasodilation. In asthmatic patient, at the lung level, it causes bronchoconstriction, and it increases the capillary permeability. This angiotensin is another peptide which is formed in the plasma from alpha 2 globulin known as the angiotensinogen. So angiotensinogen actually is a protein, and angiotensin is being cleaved from angiotensinogen. It is a octapeptide. So protein angiotensinogen is a protein, and angiotensin is, as I told you earlier, that peptides can be formed from the cleavage of the larger proteins. So when this angiotensinogen undergo cleavage, it will give you angiotensin, which is a peptide. Now kidneys, the major function of the kidneys is to maintain the fluid levels within the body, and they are also the regulator of this pH. Kidney has a role in the maintenance of pH, as well as kidney has a role in maintenance of the blood pressure. So what happens, the juxtaglomerulus cells of the kidneys, they sense the decrease in the blood pressure, and they release a enzyme that is called as renin. Renin is a proteolytic enzyme. Angiotensinogen, as I told you, it is a protein. It is a alpha 2 globulin. It is a protein, and renin is an enzyme. So renin is secreted by the juxtaglomerular cell of the kidneys in response to the decreased blood pressure. So this renin acts on angiotensin, and then it causes its cleavage and release angiotensin 1. So this picture will give you the whole understanding of this angiotensinogen mechanism. And renin angiotensinogen mechanism is a mechanism which plays role to maintain body's blood pressure and maintain fluid levels within the body. So whenever there is a decreased arterial pressure, juxtaglomerular cells, they sense that decrease in the blood pressure, and they release enzyme renin. Angiotensinogen, gin is a protein, renin and it is a substrate for enzyme renin. It cleaves a 10 amino acid peptide chain that is called as angiotensinogen 1. Then this angiotensinogen 1 is a substrate for another enzyme that is called as angiotensinogen converting enzyme. It is not only present there in the kidneys but also in the lungs and the plasma. So this convert angiotensinogen 1 into angiotensinogen 2. Angiotensinogen 1 has got 10 amino acids. It is a decapeptide. When it converts into angiotensinogen 2, the two of the amino acids are removed and it is converted into octa peptide. And this angiotensin 2 acts on various receptors on the kidney and also on the arterial walls of the blood vessels. So when it acts on the kidneys, there it causes the retention of sodium and water. When the water is retained, sodium is retained, definitely the blood pressure is being raised. And vasoconstriction is another contributor of maintaining the blood pressure. And as a result of these two effects, accumulation of, not accumulation but retention of sodium along with the water, and on the other vessel, the vasoconstriction of the blood vessel, the blood pressure will be increased. This is another picture which will give you the more clear view of this, that this angiotensinogen, a protein, renin acts on this, converted into octapeptide, that is a 10 peptide chain of 10 amino acid. Then this enzyme, angiotensinogen converting enzyme, that is called as in short ACE, will convert it into angiotensinogen 2, which is 2 amino acid less, that is it contains 8 amino acid. And then this angiotensinogen 2 also acts on the adrenal cortex. From there it causes release of the aldosterone. Aldosterone is the sodium retaining hormones. It maintains blood pressure in the body. The target tissue for aldosterone is kidney. So you can see here this aldosterone causes sodium retention. So since the sodium has positive charge and another cation, potassium, also has a positive charge, so sodium is retained, less amount of sodium is excreted, but you can see this in contrast with that potassium excretion is increasing, so that the charge ions, they are being maintained within the body. As we had discussed earlier, this renin-angiotensinogen system plays an important role in maintaining the blood pressure within the body, and we have taken the advantage of this. So ACE, that is the angiotensinogen converting enzyme, convert angiotensin 1 into angiotensin 2. So drugs, there are a list of drugs which block the activity of this enzyme ACE, and they are called as ACE inhibitors. And as a result of this, what will happen? The angiotensin 1 will not convert into 2. So when it is not converted into 2, it means aldosterone release will be blocked. So this is being used as a antihypertensive drugs. Now remember that this enzyme inhibitors, they are the commonest mode of more than 50% of the drugs which are being used, they are enzyme inhibitors. So ACE inhibitors, they are being used as an antihypertensive drugs. Another example of this peptide is vasopressin, which is also called antidiuretic hormone, and it is a nona peptide. Nona means 9 amino acids, comprised of 9 amino acids, and you can see it is unbranched, but because of the 2 cysteine residues, I am not saying cysteine amino acids, 2 cysteine residues, because when the amino acids are linked in peptide linkage, they are called residues. They are forming a disulfide linkage between the two sulfhydryl groups, and this hormone is synthesized in the hypothalamus, and it is released and stored there in the posterior pituitary, and whenever there is an appropriate stimulant, this hormone is released from the pituitary of the brain. Oxytocin is again another this nona peptide. Again, like ADH, it has 2 cysteine residues, and a disulfide linkage is being formed between the 2 cysteine residues. Similarly, it is also being released from posterior hypothalamus into the posterior pituitary, and the main target is on the breast tissues. Thyrotropin releasing hormone. Thyrotropin releasing hormone is a tripeptide and it is being released from the hypothalamus. From hypothalamus it is released and uh, its target is anterior pituitary gland. So far we discussed vasopressin and oxytocin, they were posterior pituitary hormones. Thyrotropin is released from the hypothalamus and its target is anterior pituitary. Okay. And thyrotropin releasing hormone, when it acts on the anterior pituitary, from there it causes release of the thyroid stimulating hormone. This thyroid stimulating hormone then acts on the thyroid gland and from there it causes release, synthesis and release of the T3 and the T4, which are called as the thyroid hormones. So you can see important hormones, they are being present in the category of the peptides. This picture is showing you that from hypothalamus we have 6 different type of hormones are there which are released from the hypothalamus, and those hormones which are released from the hypothalamus, they are called releasing hormone. Growth hormone releasing hormone, thyrotropin releasing hormone, or corticotropin releasing hormone. And from hypothalamus they target either anterior pituitary or the posterior pituitary. Now in this picture if you see, those hormones which are being shown with the green arrow, they are stimulatory hormone. They stimulate the release of second category of the hormone from the anterior and the posterior pituitary. Thyrotropin releasing hormone not only acts on anterior pituitary and cause release of the thyroid stimulating hormone, but it also have a stimulatory effect on the release of another anterior pituitary hormone which is called prolactin. But you can see that red arrow shows the inhibitory influence. Dopamine, which is a neurotransmitter, it inhibits that, and dopamine is released from the hypothalamus, and dopamine is a prolactin inhibiting, prolactin release inhibiting this is a substance. Now we will discuss a few of the polypeptides. If you recall, insulin and glucagon, they are being included among the category of the polypeptides. Now pancreas has a very important role in the metabolism of our nutrients, especially macromolecules, proteins, lipids and carbohydrates. And this is a picture of the islets of the Langerhans, that is the pancreatic cell. And pancreatic cell has 4 different type of cells: alpha, beta, gamma and the delta. The alpha cells of the pancreas, they release hormone which is called as glucagon, and this glucagon, the stimulus for the release of the glucagon is hypoglycemia. When individuals have less blood glucose concentration, either between meals or when he or she is in a state of starvation or prolonged fasting, then the insulin, pancreas, the alpha cells of the pancreas, they release glucagon. And this glucagon is also called as lipolytic hormones. It not only acts on the body stores of carbohydrates in the form of glycogen, but it also causes catabolism of the glucogenic amino acids because the body is in a deficiency of glucose. Hypoglycemia is a stimulus for glucagon. And so glycogen, is stored, excessive amount of carbohydrates in the well-fed state, they are stored in the muscles and in the liver. And when there is a need between meals or when the person is in hypoglycemia, body mobilizes those stored carbohydrates, and the process called glycogenolysis. So glycogenolysis is a process by which the glycogen releases glucose into the circulation. Number two, you are very well aware that there are certain amino acids. Amino acids are being classified on the basis of glucogenic or ketogenic. Glucogenic amino acids are those amino acids which can be utilized to produce glucose, and ketogenic are those which are utilized to form fatty acids. Or there are some amino acids are being categorized as glucogenic as well as ketogenic. And then we have again fats or lipids are stored in the body in the form of TAG in the adipose tissues like anterior abdominal wall, buttocks, and so when individual in a hypoglycemic state, this glucagon mobilizes glucose from the stored glycogen. As a result of glycogenolysis, it catabolizes glucogenic amino acids as well as it utilizes lipids as a alternate source of energy. We have just discussed the hormone glucagon, which is released from the pancreas. Similarly, insulin is also a hormone which is released by the beta cells of the pancreas. And the indications for the release of insulin from the beta cells of the pancreas is hyperglycemia, exactly opposite to that of the glucagon. Glucagon is released when the blood glucose level is decreased, which is called as hypoglycemia. And insulin is released when the person having high blood glucose level, that is called hyperglycemia. So when insulin is released, all those functions which are being carried out by the glucagon, they are being blocked. So you can see here that insulin stops gluconeogenesis. Gluconeogenesis is the synthesis of glucose from the non-carbohydrate sources. As I told you just before, that glucagon utilizes glucogenic amino acids to produce glucose. Okay, so gluconeogenesis stop. Number two, glucagon mobilizes glucose from the liver stored liver glycogen by glycogenolysis. अगेन दिस ग्लाइकोजेनोलाइसिस is stopped क्योंकि अभी पेशेंट की ब्लड ग्लूकोज लेवल हाई है, he or she is not in need of glucose, so ग्लाइकोजेनोलाइसिस भी स्टॉप हो जाएगी, ग्लूकोजन का अमाइनो एसिड कैटाबॉलिज्म भी स्टॉप हो जाएगी, and body will not use lipids as a source of energy because we have abundant glucose in the blood. And ketone bodies, now ketone bodies are derived lipids and they are the alternate source of energy when the carbohydrate stores are depleted. Remember one thing, when body doesn't get glucose for energy purposes, the alternate source of energy for brain and muscles is ketone bodies. So ketogenesis will also be stopped. प्रोटीन ब्रेक डाउन भी स्टॉप हो जाएगी क्योंकि जब आपके पास कार्बोहाइड्रेट अबंडेंट अमाउंट में है देन देयर इज नो sense of mobilizing proteins because proteins are mainly mean for the bodybuilding purpose, they are not the primary source of energy. Those functions which are being promoted by the insulin is that क्योंकि ब्लड में ग्लूकोज लेवल हाई हो गई है, नाउ द फर्स्ट मेजर फंक्शन ऑफ इंसुलिन इज टू नॉर्मलाइज दैट ब्लड ग्लूकोज लेवल. सो व्हाट इट विल डू, इट विल कॉज entry of glucose from blood into the cell so that the blood glucose level should become normal, number one. And once the glucose is within the cell, तो cell के अंदर then it stimulates glycolysis. Glycolysis is the breakdown of glucose in the cell so that the cell can now utilize that glucose for energy purposes. और जो एक्स्ट्रा ग्लूकोज है क्योंकि हाइपरग्लाइसीमिया इज एन स्टिमुलस फॉर इंसुलिन रिलीज, तो दैट एक्स्ट्रा ग्लूकोज विल नाउ बी डाइवर्टेड टुवर्ड्स ग्लाइकोजन सिंथेसिस. अच्छे वक्तों में आपके पास ग्लूकोज ज़्यादा है, आप इसको स्टोर करो एज ग्लाइकोजन. and it also it is an anabolic hormone. Anabolic hormones are those hormones which promote synthesis. तो आप देखें, it promote glycogen synthesis, it promote protein synthesis, it inhibit lipolysis. Lipolysis means breakdown of lipid. तो lipid की breakdown को block कर रहा है, protein synthesis बढ़ा रहा है, glycogen synthesis को promote कर रहा है, and it's have a important effect on minerals, that is the potassium and the phosphate. Insulin always increases the entry of potassium as phosphate और uptake of these ions into the cell. ये cell में potassium और phosphate की entry को enhance करता है. So these are the functions of hormone insulin, which is a polypeptide. Now so far we have discussed various peptides and polypeptides with their biomedical role within the body. Now we have been with the protein. Though the Dr. Amna has already been giving you the biomedical significance of protein, but we will go through briefly. We'll go through the important role which the proteins play in the human body. Now you are very well aware that cytoskeleton, which is present within the cell, and in the cell it maintains the shape of the cell, and in the human body this internal protein network maintain cellular shape and physical integrity. Then as actin and myosin filaments, they constitute the contractile machinery of the muscles. We have hemoglobin, which is being responsible for the oxygen transport. Not only for oxygen transport, hemoglobin have some other functions. It has a role as buffers. It also, beside oxygen transport, it transport carbon dioxide, and it is a transporter of the nitric oxide also. Then as immunoglobulins or circulating antibodies, they search out foreign invaders. And the most important function is that almost all of the enzymes are protein in nature. So these enzymes, they are being required for various metabolic pathways, and they are being required to generate energy, synthesize and degrade biomolecules, and also they have a role in the replication, transcription, and this protein synthesis. As receptors, they enable cells to sense and respond to hormones and other environmental cues. Beside these, in electron transport chain, they are the source of generating energy in the form of ATP. And not only hemoglobin, we have example of the hemoglobin as a transport protein, but we have this proteins acting as a carrier molecules like albumin, alpha-1 globulin, beta globulins. They all also act as a carrier molecules, transporters. Then they serve, if we have gone through this structure of the plasma membrane and cell membrane, and where we have seen that integral proteins, they act as ion channels and pumps. Ion channels for transport of ions, and pumps like sodium potassium ATPase pump, and as receptors, hormones and cytokines for intracellular communications. And they also constitute the signaling network for intracellular communications. So these are the various functions of protein in the human body. These pictures give you again the various functions of protein they serve in the body. You can see they're constituting the structural and mechanical support to the body. They act as enzyme. Many of the hormones, they are protein in nature, like we have just discussed, peptide of protein nature, glucagon, insulin. And then antibodies, they are again globulins, immunoglobulins. They are glycoprotein nature. And then as protein, plasma proteins, they maintain colloidal osmotic pressure. They act as buffer. And then we have just discussed that the integral proteins, they act as ion pumps and channels for transport of ions. And then we have this various proteins which have a role in the transport, like we have transferrin, albumin, hemoglobin. So these pictures give you a clear concept of the various functions of the protein in the body, the role of. Now, what happens that once the proteins are synthesized in the body, the process of protein synthesis is called translation. And as I told you earlier, that proteins are synthesized by the ribosome. Ribosomes are protein synthesizing factories, and while the proteins are being synthesized, they undergo folding, because the active protein is that protein which is assume a three-dimensional structure, which is being folded into a complex structure. And we have just discussed, when we are going through the difference between the polypeptides and proteins, that the proteins, beside having peptide bonds, they have some other non-covalent bonds like hydrophobic interaction, hydrogen bonds, disulfide bonds. So functional protein, they do undergo folding, and this process of folding takes place while the ribosomes are synthesizing proteins. So when they are being a class of protein is called chaperones. Chaperone proteins, they actually assist or they guide proteins to be folded in the right manner. So what happens that sometimes due to the genetic mutation or due to the errors in the translation or due to post-translational modification, what happens that when proteins are synthesized, then the certain groups are to be added to these proteins like phosphorylation of protein, methylation, acetylation, glycosylation. So if these are called post-translational modifications, so any abnormality or during the process of translation or the event which is causing defects in the post-translational modification, so protein they may be misfolded. Now these misfolded proteins, they may be non-functional, or they may not reach to their destination, or they may cause toxic. Now, normally, as I told you, that these misfolded proteins are usually degraded. But what happens in some circumstances, they are resistant to catabolism, like amyloid proteins. So when they are resistant, so they persist there, and then they can be a cause of disease, or they can cause. Among these resistant proteins, following disorders are being experienced. Uh, Scrapie disease in the sheep, mad cow disease in cattle, and human beings are being victimized by this accumulation of the non-degraded abnormal proteins or misfolded proteins, and these are called the Alzheimer's disease and Creutzfeldt-Jacobs disease. These both disorders, they involve the central nervous system, and the signs and symptoms of these disorders includes memory problems. They have been observed with the behavioral changes, there's poor coordination, and visual disturbances are being observed. If you one of the known personalities, that is the President Reagan of America, is a victim of Alzheimer's disease. So Alzheimer's disease usually affects people in the older age, and it mainly affects the central nervous system. Now this Alzheimer's disease is being caused by the misfolded amyloid beta proteins which get deposited in the brain, and these amyloid proteins are resistant to degradation. They cannot be catabolized, or so when they are being deposited in the brain, they cause neuropsychiatric abnormality. And here you can see the comparison of a healthy brain with the brain of an Alzheimer's patient suffering from Alzheimer's disease. You can see there's a shrinkage of this cerebral cortex as well as those of the hippocampus. And the cerebral cortex, as you know, that it is responsible for the language and information processes. And the third thing which you can observe, that ventricles are dilated and they are filled with the cerebrospinal fluid. So they are large, they are dilated, and they are filled with the cerebrospinal fluid. Rest of the brain, cerebral cortex is being wrinkled, it is being shrinked, and it has been contracted. So those areas which are mainly concerned with the language and with the information processing, they are mainly affected. As far as this hippocampus is concerned, this hippocampus is mainly concerned with the consolidation of information from short-term memory to the long-term memory. So again, this Alzheimer's disease is because of the deposition of the amyloid protein, which is a misfolded protein and which is resistant to degradation. Now, we have already discussed this Creutzfeldt-Jacobs disease, and we have already discussed that this disorder is also affects the human beings, and it is being presented with the poor coordination, memory problem, behavioral changes, and the visual disturbances.